Okay, can everybody see my slides okay?
Yes.
All right. Well, thanks, Carol, and thanks everybody for joining us today for the Alliance webinar. Today we're going to talk about how AI and machine learning inform literature curation at the Alliance. And as Carol said, I'll be joined today by my colleagues Valerio Arbnaboldi and Chris Tabone. So as many of you are aware, the Alliance of Genome Resources is a coalition of intensively studied model organism knowledge bases that have come together to provide a common user interface and experience for interrogating the genome and gene function for each of the respective species that are part of the Alliance. So information at the Alliance is centered largely on individual gene pages, and shown here is an example of a human gene, the AFG3L2. And gene pages at the Alliance are organized according to specific types of information. So the Alliance facilitates this cross-species analyses in several ways, two of which I've shown you here. So one is an orthology widget, where given a particular gene page, you can go and see all of the orthologs for the member species that are shown here with links out to go to the corresponding gene pages. And for comparative views, shown here an example with gene function annotation using Go annotations, where we have things like this ribbon display where you can get a sense of all of the gene function annotations across all of the species here for different aspects of Go. So in order to create the gene pages with all of this data, there's a lot of work happening behind the scenes with respect to harmonizing our information and our data models and developing common software. And the example we're going to talk about for common software today is our Alliance literature management system and the AI and machine learning that we are incorporating into that. So just to sort of give you an overview, so in addition, of course, to genome annotation, really the heart of biological knowledge-based curation is curation of the published literature. And this slide is really just meant to sort of give a generalized overview of the literature curation workflow at a lot of knowledge bases. So I want to emphasize that while there are slight differences and nuances in how the individual groups might approach this, this is generally how it works. And so the workflow here is color-coded, and this is started to indicate, again, generally the status of where these things are at the Alliance, although, again, different groups might be at different points in the automation. But generally, we start with identifying candidate papers to include in the corpus for each of the model organisms. And this is almost exclusively done with PubMed queries. Once we identify candidate papers, the next step, which is still largely a manual step for many of us, is the decision of whether or not to include that paper in the corpus for the particular organism. Once we've decided to include a paper in the corpus, we then acquire the full text of that paper, and that includes supplemental material. And then once we have the full text, it goes through steps such as indexing, some groups call it, where we'll identify the entities that are studied in that paper. And by entities, I mean things like genes, variations, strains, and species. In addition, when there are often new entities described, like new alleles, groups will create those entries in the knowledge base. For some of us, we then do an automated document classification, which really just tells us what kind of information is in the paper. Does this paper have experiments on anatomical expression, maybe protein-protein interactions? or RNAi experiments. And then there's also work that curators do to triage then and prioritize papers for curation. And that may depend on the information content for particular genes and say wanting to get new information for genes that may not have much. And then lastly, once we've done all of this indexing and classification and triage, it will go to the professional biocurators to do the actual data curation, which is really the synthesis of all of this information into data models and to present that information there on the webpage. And this you might also have heard referred to as fact extraction. So our goal as part of the Alliance Literature Group and AI Working Group is to get as much of this process as we accurately and reliably can to be green and blue, which means that we want to employ as much AI and machine learning as we can to help us be as efficient as possible in this literature curation workflow. So to do this, we have developed a system known as the Alliance Bibliography Central, or the ABC. And this is a unified literature management system that has been developed from the ground up to be integrated with state-of-the-art AI and ML. So right now, this is still an internal tool to the Alliance, although we would like to, in the future, offer some of the capabilities of this more widely on the Alliance website. But what I'm showing you here is in our production tool, the search interface that Alliance curators can use to go in and, based on the information from our classifiers and our entity extraction and where papers might actually be in that workflow, they can come here and use our faceted search to find the high priority papers for them to curate. So as I've said, in this interface, we have information about the workflow tracking. We have information about the different topics or data types and entities that we've associated with papers. To do that, we've made use of ontologies. One ontology we've developed internally is known as the Alliance Tags for Papers, and that has terms for our workflow, our curation status, the topic and entity tags. And then we have also had a nice collaboration with the evidence and conclusion ontology developers so that we can track the provenance of where these topic and entity tags and workflow tags come from. In addition, we have developed extensive data models for storing all the information about the AI and ML methods that we use. We've developed a manual validation interface for professional curators to come in and look at all the results of our AI and machine learning and validate things as positive and negative. This is extremely valuable for us to, while we do this work, continue to create training and testing data for models and keep that as up to date as possible. And then coming soon, we will also be developing a more extensive training set manager that curators can use to create and refine training data for all of these tools. So I just wanted to show you what this actually, this validation actually looks like in the ABC before I turn it over to Valerio and Chris, who can tell you more of the details about the methodology. So this is a view of our table that shows what kinds of information has been associated with the paper. This is taken from a paper on C. elegans. So you can see we've associated that with the species C. elegans. And these are a number of the document classifiers that we currently have in production. So it will tell you what kind of data is predicted to be in this paper. We also have predictions for data that is not there. So you'll see that in this confidence level column, it says negative, and we predicted no data. This paper is predicted with a medium confidence level to have catalytic activity. And I, as a professional curator, can come in, and I've already done this and validated that this is correct. And the machine learning got this correct. And other curators will be able to come in and do the same for data types that they might curate. So right now, this is independent of curation, but I also want to mention that we would like to integrate this validation with curation so that curators don't necessarily have to do this separately. If they have curated a particular data type, that is validation that the machine got it right. So again, this is a really nice interface and a way for us to easily validate the results that we get. And not shown here, but an option that we also have, another column we could show, is the results of community or author curation. So as many of us rely more and more on community curation, we will have the results of that here as well. So we can compare the machine learning with the professional biocurators and with the authors to help make that pipeline as reliable as well. So with that, I am going to turn it over to Valerio, who can tell you a lot more about what's happening behind the scenes with these different methods.
Thank you, Kimberly. Let me share my screen. Can you see the slides?
Yes.
Yep.
Thank you. So my name is Valerio Arbnaboldi. I'm a developer at the Alliance. I work on the ABC. I'm also part of the Worm-based team, and I'm the working group lead for the Alliance AI and Machine Learning Working Group. At the ABC, as Kimberly already introduced, we're using several machine learning models to automatically extract information from the full text of publications. For example, we have the document classifiers that are able to identify different data types of interest, such as physical interaction, human disease, and many others, from the full text of the papers. For this specific models and for the current implementation we have in production, we're using supervised learning models, such as SVM, neural networks, and others. And we train these models on manually labeled data sets collected by professional biocurators. At the moment in production at the Alliance, we have 19 models for different mods, part of the Alliance, and we are constantly working to train new models. These types of models can be binary classifiers, able to tell you whether a specific data type is present or not inside a publication, but can also be multi-class. For example, we're using document classifiers to sort publications into different classes of priority to help curators in the pre-triaging and triaging processes. We also have named entity recognition models that are able to tag publications with a list of biological entities, for example, genes, species, alleles, and others, extracted from the full text of the papers. For these specific types of models, we're using string matching and regular expression on Alliance entity names as much as possible, especially for organisms like C. elegans, where the gene names are very easy to identify in the full text, and the nomenclature is well-defined and very unique. In other cases, for organisms where entity names are harder to extract, for example, fly-based and fly genes, we use language models such as fine-tuned BERT models and other large language models to extract the information from the literature. For this type of models, the named entity recognition, we use them for identifying both existing entity names that are already present in the Alliance database, but also for potentially new entities, especially using regular expressions. We also have sentence classification models that are able to extract sentences of interest for curation from the full text of publications. We're using these models, for example, to help professional biocurators or community curators in focusing on parts of the publication where we really know that there's curatable information. all the information that we extract from the publications are attached to the references and shown to the curators in the ABC. But we're also using all the information to index publications. And these are included in our search engine for literature. This way, curators can find publications of interest by searching for entities or data types that are interesting for their data curation. And also, all the information is provided to the curators so that they can use, for example, the sentences that are identified in full text as curatable, they can be used by the professional biocurator as additional evidence for their curation. Another AI project that we're working on at the Alliance at the moment is the generation of gene summaries using LLMs. Currently, the Alliance is generating gene summaries automatically using an algorithm that uses curated annotations for the genes and generates gene descriptions for different data types, including the gene ontology, disease, expression, and other. This algorithm collects all the annotated terms from ontologies to the gene and then creates a human-readable, concise description that conveys the main functions of the gene. When the list of ontology terms is too long to be included in a concise description, we use an optimization algorithm combined with the ability to traverse the ontologies to select the best combination of ontology terms to be included in the file description to maintain readability and provide as much information about the gene as possible. The limitation of the current implementation is that we can only generate gene descriptions for curated genes. And for genes for which we don't have curated data, there's no automated description available. For this project, we want to try to use LLMs to automatically extract unstructured information from the literature and generate gene summaries directly from this information without relying solely on curated annotations. So the pipeline of this project is divided in 2 steps. In the first step, we use an LLM to automatically scan the literature and extract information in a structured way related to the genes of interest. For this, we're mapping ontologies as much as possible to provide reproducible and correct output. Once we have this extracted information from the publications, we in the second step, we use another LLM to generate a concise summary for the genes based on the extracted information. And also in this step, we're using structured output as much as possible and a very precise prompt to make sure that we're controlling as much as possible, reducing the hallucinations and having as much reproducibility and correctness in the final generated description as much as possible. And next, my colleague Chris will talk about another project related to AI at the Alliance. Thank you.
Okay, thanks, Valerio. Let me just share my screen here real quick. Okay. All right. So my name is my name is Chris. I'm a senior programmer, analyst, and developer at the Alliance, and also I work with Flybase. I'm employed at the Jackson Laboratory. So, oh, I should ask, can everyone see my screen? Does it look okay?
Looks good, Chris.
Okay, thanks. All right, so I'm just going to spend a couple minutes here talking about kind of an LLM adventure that we have going at the Alliance we call it the BioCurator Assistant. And it started out as kind of a proof of concept last year, and it's grown into a tool that we're going to integrate into the ABC literature system and hopefully use throughout the project. And what it is a command line Python-based program that uses LLMs, and specifically we focused on OpenAI's GPT series because that was one of the first ones widely available with a good API access. And it lets us pass it. We pass it publications, full PDFs, and then we use it for data extraction and for triaging. So it started at Flybase early last year. We moved it to the Alliance. The code is publicly available. There's the link. It's in our GitHub repository. There's going to be a rework in the next few months to update some of the API requests because OpenAI is sunsetting some of the other older assistance approach that we're using. But it's under active development and it's available there if anyone's curious. Take a look. So what it basically does, as I mentioned a minute ago, is we use it for publication triage, wherein you present it with a publication in PDF, and then it can tell you basically what kind of data is in the publication in an overall sense. So does this publication have new alleles? Does this publication have new transgenes? Does it contain disease models? We ask these questions through customizable prompts, as I'm sure many people now are familiar with. The assistance API that we're currently using lets you add specific set of instructions aside from the prompt to tell the biocurator assistant, you are an expert biocurator, this is your task, and so on and so forth. One of the benefits we find from our approach is that versus older approaches like the SVMs or other models is that we can request the reasoning for the classification to be returned to be reviewed by the curator. So if the program makes a good call and it says, this paper contains a new allele, and then it provides a justification. If it gives us a false positive or a false negative, we can see the reasoning of the LLM in this sense, and we can then use that to iteratively improve our approach into for further curation. And then besides the publication triage, the biocurator assistant also works for some named entity recognition. So in addition to classifying the papers that contain specific types of data, we're also asking it to extract the actual entities from the paper. And we do this with a structured JSON output so that we can pull the entities out and then sort them and organize them in a way that's easily usable for our programs or for curator review. And we've tested this with some data from Flybase and Wormbase and a little bit from Zfin as well. And just to give a quick example of what the extracted data looks like. So in the left column here is the names of things as written in a publication. The middle column here is this is a Flybase paper. So this is how a curator at Flybase would classify these things. And then on the right is this is O1 Preview. Is actually an older model now because new models are coming out all the time. But this is what it was able to extract from the paper. And we just gave it the publication. We didn't give it any extra data, and we tell it to focus exactly on the publication. And it does an impressively good job at entity extraction. And we find the same with triage. So it performs as good as or better than a lot of our older models. And the goal in all of this is to present it through the ABC system in a way that's easily reviewable by curators before it can go into the database. So the idea that you're curating a paper and you're presented with a screen that says, these are the types of data in the paper. Here's the genes and the alleles and other things that we think that are in the paper. And then you as a human, as a curator, review that before it goes into the database. And we have a lot of work to do on it. It works very well now. Again, feel free to check it out on GitHub. We're going to benchmark some of the newer models that are coming out, and we need to set up a kind of an iterative feedback line for prompt optimization along with the curators, especially when we get these false positives or false negatives. And we're looking into retrieval augmented generation. So we want to tie in ontologies and things of that nature to actually create complete annotations. So extract the data and then have the LLM help us assign annotations appropriately using ontologies and using the data that it extracts. And then again, this year we'll have it hopefully integrated into the ABC workflow. And then we're going to expand it as much as we can to see what the limits are, what kind of data can this extract, what kind of entities can this extract. And the one downside I do want to mention, which I didn't put on the slide here, but it works very well right now, but there is the negative aspect is that it's a proprietary algorithm, proprietary LLMs. So it could change at any time. We don't have full control over the API that OpenAI is developing. There are local models like Llama and other ones that we could use. We don't currently have the hardware required to run those yet, but that's more of a grant funding issue. So maybe down the road we could try some non-proprietary open source LLMs, which could hopefully provide us with similar results. So with that, instead of sharing the screen back again and getting all chaotic, I'm just going to move on to the acknowledgement slide. So these are a list of the people that have worked on everything that we've talked about, the blue team and the literature and AI working groups. You can see everyone listed here. And then we're thankful to the NIH for the funding of this project and everyone else that's listed. So I think with that, we'll open for some questions.
Great. Thank you, Valerio, Kimberly, and Chris. We do have time for questions. So I'll open it up if you want to put your question in the chat. We also have the Q&A tool. And I guess I'll start. So Kimberly, at the beginning, you mentioned the topic tags that get attached to different papers. So if I wanted to add a new tag, let's say I want to, all of a sudden I want to tag publications that have rescue experiments in them, and that hasn't been a tag for many, many years. How long would it take? Would you retrospectively go through the literature and try to find ones that were relevant to that tag? And if so, how long would that take?
Yeah, that's a great question. So absolutely, if a curator needs to create a new topic tag, we would add that to our ontology. And Carrie Van Slyke, who's been our product owner of the Blue development team, has been doing that. And so, yeah, it's a good question. What do we then do, right, with legacy literature? So I think that if we have positive and negative training set papers that we could use for that, we could absolutely go back and take the full text of what hasn't been classified for that data type and do that. And we've actually done that with training some new classifiers for some of the C. elegans papers. So absolutely, that's possible. Yeah.
Great. There's a question. Oh, go ahead, Valerio.
Yeah, sorry. I just want to add that the way we created the workflow manager at the Alliance is that we attached tags to all the papers that we want to classify to say this paper needs classification. So you can even be selective and say, I don't want to go back and reclassify the whole literature, but maybe you can find a subset of the papers that you want to classify. You attach the classification needed tag for the new data type and do the classification on those. So there's a lot of fine tuning that we can do at the APC next to this new workflow manager that we're using.
Fantastic.
Yeah, we have that flexibility built in. Right?
A couple things in the chat. And was your comment related to this topic?
I'm not quite sure. Go ahead with the others first.
Okay, so a couple questions in the chat. One question is about redacted publications or information. How would or could that be addressed in this system?
Yeah, so absolutely, we will deal with retracted publications, and we have had some very recent discussions about that with the Blue team, and so we want to make it very clear once we know that a publication has been retracted, that the curators and the system knows about that. And we're in discussion about then what to do exactly with the flags and tags that go on that paper. I think we'll probably sort out some of those details very soon in the coming weeks. But bottom line is that, yeah, absolutely, we're aware that we're going to have to deal with that.
Chris or Valerio, any comments on that topic?
Yeah. The workflow manager, we talked about it yesterday, but the idea is we will create a special status for the paper that is retracted. And once we transition in the workflow to that specific status, we can then call different actions that will in turn either delete the automatically extracted information or maybe keep it if it has been validated by a curator. So depending on the use cases, also in this case, we can fine tune the workflow and decide what to do depending on the type of publication and the status, specific status.
Great. Next question. I noticed in your data type list, you didn't include phenotypes other than knockdown, overexpression, or go biological process. These are probably more difficult, and at least for palm base, the largest and most difficult extraction problem. Any progress here?
Yeah, Val, are you still here?
Val's, I think Val's.
I am, yes, sorry.
Okay. I just want to clarify your question before I answer it. So, what other, so we have RNAi and variation phenotypes and overexpression phenotypes. Is there another kind that you were interested in?
Well, just any, I'm thinking more of I'm thinking less of the mechanism of creating the phenotype, but just more generally identifying phenotypes that are just engineered, perhaps an amino acid mutation that's engineered, or so just
You mean like a CRISPR kind of thing, or?
well, we have them, we have thousands of them where somebody will have just engineered using some transformation, just engineered specific amino acids, not just knocked out. And Cerevisiae will have the same, where actually I don't think they curate that detail always. I think they may do it in notes, but all our alleles are most of those aren't knocked down or overexpression, put it that way, unless they're either a complete deletion, which I suppose is close to the knockdown. So I guess it would identify a deletion, or that could be done easily, but it's more, yeah. So are you looking for phenotype terms? Because all the other ones are sort of, you're looking for active. You're looking for the data type. I see. It's just for the data type rather than a specific term to extract.
Yeah, so the document classifiers that we have right now, at least the ones that we have been using at Wormbase for a long time, are just at the level of a particular data type, right? So like this paper is likely to have an RNAi experiment. This paper has anatomical expression. We have done and are doing more intensively, the sort of more detailed, like as Valerio mentioned, sentence classification for the purposes of fact extraction, right? And so, which case we would take that information that we can get from those sentences and then try to do our best to translate that into like on matching ontology terms, right, that a curator would then use to actually make the annotation. So that work is ongoing. Yeah.
So presumably you do manage to capture most publications that have phenotypes, but what about biological process? Because that wasn't in the list.
So we don't and have not tried to classify papers for Go biological process, right? With the thinking that for curation, at least for C. elegans, right, things like phenotypes are often what we use to inform whether or not we'll make a biological process. So we do sort of the data type rather than we're going to look for, papers that might be suitable for Go biological process.
Yeah, that makes sense, actually. Yeah.
Yeah.
Because you use all the other information to
Exactly.
make that
Exactly.
decision.
Right.
Okay.
Great.
Yeah, thank you.
Anything else, Val?
No, that's good. Thanks.
We'll take one more from the chat and then we'll go to Anne and then we'll go back to the chat. So will the gene summary generated for those genes without annotations be tagged without curator review?
Yeah, thank you for this question. Right now, we're at the stage where we're reviewing the results of this newly LLM generated gene summary. So we haven't, we're not publishing these gene summaries on the website yet. And I want to thank Ranjana Kishore and Pengyuan Lee who are working with me on this project. We discussed this. We think that yes, we'll need to provide some statements, making sure that the users know that these are LLM generated gene descriptions, and there could be hallucination or inaccurate facts. But from the results that we've seen so far on a subset of genes related to Alzheimer's disease, so this description we are evaluating were specifically related to Alzheimer's disease, we found that the genes that had no description from the annotated information are gaining some descriptions that they actually make sense. So we're pretty confident that we can publish this new LLM generated gene summaries on the website. But yes, with disclaimer that there might be inaccurate data.
Thank you, Valerio. Anne, would you like to ask?
Sure. I'm not sure this is exactly related, but you know, when you run the ABC, it relies on papers already being deemed in a corpus. Is there any possibility of just being able to upload a PubMed PMID and have this run on, you know, a general paper that somebody would want? And I think this is maybe related sort of to the next comment or question in the chat.
So is it more of an on-demand kind of processing, Anne? Is that what you're thinking of?
Yeah.
Well, we have an upload feature for the mods that are part of the Alliance. So if you don't have a specific reference in your corpus, you can add it to your corpus manually. Or we have a bulk, what we call the bulk uploader, where you have a folder with all the PDFs or your publication following a specific nomenclature. You can upload them in bulk, which is what we do at Wormbase. Then they are automatically included in corpus for your mod. But we don't have a way at the moment to create a new corpus for a group that is not a mod at the Alliance. But in theory, the same methodology that we're using for all the mods could be used to create ad hoc on-demand corpora for the users. Right now, the system is only for internal use by mods curators at the Alliance. So we designed that around all the different mods and their corpus.
Thank you.
Okay, we'll go to what I think Anne was referring to as the related question. What are the technical resources needed to run ABC? Is this system something that could be deployed for other resources? I'm thinking about the many plant and animal ag databases that are part of the Ag Biodata Consortium that would be blown away by this amazing system.
Yeah, I can answer this question. Everything runs on AWS and all the services are in Docker. So it's easy to deploy the same infrastructure on in another place. And we're not, the AWS instances that we're using are relatively small and they are expensive, but it depends on the amount of data that you want to index. At the Alliance, we're indexing more than 1 million papers, and we have a lot of information that we extract and we send into the database. The other aspect that is very important and takes up much of the resources we're using right now is the automated indexing. So as soon as we upload a publication to the ABC, or we extract information from that publication and we send it in our database, this information is automatically synchronized in our search index so that it's available right away on the fly for curators in the search interface. And this mechanism is could be deactivated if not important, but it's taking up, let's say, 30% of resources that we're using.
And we also, and I'll chime in for a second, there's also some tie into Okta for authentication, and there's tie into some GoCD continuous deployment services that we use. And so there are some like scattered resources for the ABC, especially for the LLM stuff that we're doing right now. I mean, personally, I think it would be a fantastic resource for the community to make this deployable, installable package, or at least provide detailed instructions on how people could set up their own. I mean, there's been wild success with things like Chado or some of the Go tools or the Genon, like that. It's great to see that stuff adopted. I think there might be a question of resources right now at the moment. We're trying to get a lot of the mods fully moved over to the Alliance. But that being said, I mean, we'd have to speak with the PIs and see if they think that's a viable path to go down later, so that that like AgBio could just take this, deploy it, and then use it for their own set of publications. I think that'd be fantastic.
It's also very modular. So depending on the services that people need, they can deploy just the services that they need for their specific triaging or curation purposes. It doesn't have to be deployed all in one big package.
Great. So there's a question about mouse. So the vast majority of the literature about mouse is not relevant to the mouse genome informatics database. And MGI currently uses a support vector machine learning approach to designate papers that have potential MGI relevance, and it assigns a confidence for each call. And the question is about, does the Alliance use its support vector machine and or AI models for a similar binary classification of journal articles about Alliance relevant or not?
This, I mean, you use in or out of corpus for a particular mod organism. Is that right, Richard?
Yes, yeah, it would be for each organism, but together you could collectively consider it Alliance relevant if it's relevant for any of the organisms, right? I mean,
Yeah.
I guess the point is, as Carol was saying, the vast majority of published mouse papers we don't even want to spend time looking at, right? And
Right.
so, and I assume it's got to be similar for maybe not as much for some of the other mods, but you must encounter that, and I'm sure that your current models will, basically not classify it for anything that is useful for you. So, but to have a high level first pass analysis to say, okay, don't even bother sending it to the rest of the system because it's very unlikely that we're going to use it. I'm just wondering if you've looked at that yet. And I know the AI is not really particularly designed for binaries, but you could probably train it for that, right? So anyway, just wondering.
Yeah, no, we've definitely talked about that as well. And you're right, there are real differences amongst the different organisms of, can you, based on an abstract, right, make a quick decision of whether or not a paper is going to be in or out of your corpus. And for some groups, right, that's a lot harder than others.
Yeah.
But we have definitely talked about, and as I sort of mentioned in the workflow slide, that, wherever we can accurately, introduce AI or machine learning into the very, decision points there, we would like to do it.
Yeah.
Yeah.
We certainly can't do it from abstracts alone.
Right. Right.
But, just a couple of things, just real quick. Jim Caden developed this, but we found what's really, really informative are things like figure legends, that kind of thing. So, and anyway, certainly if you ever get to the point where you're looking into it, we're happy to help out with that. So.
Yeah, great. That'd be good.
We have one more question in the chat that sort of goes back to the question asked earlier about retracted papers, but it's really a question for each mod rather than about the ABC system here. And it's, does any model organism database have standard pipeline to follow retracted papers and remove the annotations from the mods? And I think that's a per mod type question. I don't know if Chris, Valerio, and Kimberly can answer that for all mods.
Well, what I can say is that we're thinking about setting up a centralized workflow management for this at the Alliance, but each mod can then customize their actions. If a paper is retracted, for example, Wormbase would like to remove all the related annotations. Maybe another mod would like to remove automatically extracted information, but not the manually associated entities or data types. And these actions are customizable for each mod at the Alliance. This is the idea. So we want to provide a harmonized view, a harmonized workflow management, but at the same time, we want to give the flexibility to each group to define their own actions, and that can be slightly different, but the overall workflow will be similar.
Excellent. I don't see any further questions in the chat. I just want to thank Chris, Valerio, and Kimberly for a fantastic presentation, and everyone online for the questions and interaction. This session, like all of the others in our office hours series, will be posted on the Alliance website so that it can be viewed asynchronously. And our next Alliance office hours next month is going to focus on the use of JBrowse as a genome browser for multiple model organisms, and hope to see many of you at that office hour session as well. So thank you very much, and we hope to see you again.
Thank you.
Thanks, everybody.
Thank you.
Thank you.